This Week in Virology, the podcast about viruses, the kind that make you sick.
From Microbe TV, this is TWiV, This Week in Virology, episode 954, recorded on November 11th, 2022. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello, everybody. It's raining outside, so this is the storm that's moved up from Florida and is now drenching the Appalachian Mountains and causing massive flooding and downing trees and knocking out electrical systems. And it's just a big mess out there right now, but I guess we could use the water, so never refuse rain because you never know when the next time it might rain.
Well said, Dixon. I like that. Also joining us from Madison, New Jersey, Brianne Barker.
Hi, great to be here. Unsurprisingly, I have basically the same weather as Dixon. It is raining here too. 63 Fahrenheit, 17 Celsius. Good day to be inside.
From Austin, Texas, Rich Condit.
Hi there. 53 degrees and cloudy. It was, we have a really steep cold front coming through. It was a couple hours ago, it was 20 degrees warmer, right? And we've gotten much less rain with this than, it was really disappointing. I was hoping for a real show, but nah. It's all good.
So it's funny. I so associate you with Florida. I was going to ask you how you did with the hurricane.
Well, I have friends who have coped with it, including my brother. You know, he's in the Jacksonville area on the coast. And then my sailing buddy, Henry Baker, has a beach house near St. Augustine, and that's where he keeps his boat. They came through okay, but if you look at what happened to the beach, there was some pretty significant erosion.
Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 64 degrees, and but the Norwegian weather app says it's 16, which would mean it's 61. So it's somewhere between 61 and 64. But I'm going to tell you all these temperatures in Fahrenheit. So, earlier in the week, the forecast was for yesterday to be 70 and today to be 71. Now it's going to be a little cooler than that. But from that, we also knew that tomorrow it's going to be in the low 40s with lows in the 20s. So that same cold front probably that Rich is getting, we're getting with little snowflakes on one of the weather apps for Sunday. So.
I think this sounds pretty similar to what they're talking about for us.
Yeah. It's all McFadden's fault.
And at those interfaces, you get all the kinds of crazy weather like tornadoes and et cetera, et cetera.
So let's find out what the weather is in the northwestern part of the U.S. Our guest today is from the Washington State University Pullman, Michael Letko. Welcome to TWiV.
Yeah, pleasure to be here. Thanks for having me. So I guess I could share our weather. It's sunny. It's bitter cold. It's starting to really turn into winter already. It's about, 3 degrees Celsius outside. So it is 38 or so Fahrenheit. So it's winter's already getting here as it turns out. Right. We have snow projected for the next couple of days. So.
But you guys live in a semi-desert area. Is that correct? So you don't get a lot of precipitation usually, do you?
The Palouse, where Washington State University is, gets a fair amount of annual precipitation. So we're one of the nation's largest wheat growers. So wheat and legumes.
You called it the Palouse?
The Palouse, yeah. It includes basically this whole area of southeast Washington, right on the border with Montana and Idaho, and getting kind of right on those Rocky Mountain area.
I'd never heard that before either, Brianne.
I was interested in the biome as well, Dixon, and actually looked it up. It's described, I think, as prairie, really.
Yeah.
Is that fair?
Yeah.
Mm-hmm. Interesting. I wouldn't have guessed.
Big rolling hills.
And
rolling hills here.
Palouse is spelled Palouse, right?
Yep.
Yeah.
Cool. All right. Before we
it's a beautiful area, by the way. I've been there. It's just gorgeous. I love where you are.
Yeah. Before we start talking with Michael, I have a couple of announcements. First of all, as many of you know, over at vaccinated.us, they sell Spike T-shirts. And starting in September, they've been doing a fundraiser where you go buy a Spike T-shirt. They give the profits to Microbe TV. Very cool. And that worked out really well, so they extended it through October. They have different color shirts, men, women, kids shirts. And now they're extending it to the end of the year. So if you want a Spike shirt, they're very cool. The spike protein of SARS-CoV-2, what they call the molecule of the year. Buy a Spike shirt, wear it, show your love for Spike, and support science education here at Microbe TV. You just buy your shirts, you put them in the cart, you go to checkout, you use the promo code Microbe TV, and that will direct the profits to us. Thank you so much.
Has anybody thought of getting Spike Lee to help spur this on?
Probably be too expensive.
He might actually be willing to do it.
Yeah, but it'd be expensive because he'd just think we're
for a good cause. It's for a good cause.
I know it is. Look, I can't even get in touch with Malcolm Gladwell. I'm never going to get in touch with Spike Lee. Okay?
Oh, really?
So Malcolm Gladwell.
I really like my Spike T-shirt, by the way, and it's a good education opportunity, because it's really pretty. And and so I tell people, I ask people, do you know what this is? They say no. I say, well, it's the coronavirus spike protein, which means something to everybody. Yep. Looking down at the top, I point out the threefold axis of symmetry, and they've really stylized it really nicely, and it makes the point that biology is really beautiful.
We actually talk about spike protein today. Before we do, all right, so that's our fundraiser. And then I mentioned Malcolm Gladwell. So Malcolm Gladwell, we understand you like TWiV. You listen. We'd love to have you on. So I wrote your publicist, crickets. So get in touch with us, okay? This is a good cause. It would be a lot of fun to have you on. And just TWiV at microbe.tv. And then finally, this is the time of year when we do our fundraising here at Microbe TV, end of the year. And why we do that is because it's a time when people need to give away their excess money, which we appreciate. And so we have a special deal for you this year. Last year we gave away copies of Principles of Virology for a donation of $1,000 and more, and it was signed by me. And that's okay. We got some good donations, and I appreciate that. But this year, we're starting to work on the 6th edition of Principles of Virology. So we got together not too long ago, and Jane Flint said, I have 10 extra copies. You can have them. And I said, okay. She said you could give them away. I said, everybody's here. Why don't you all sign them? So I have 10 copies of Principles of Virology signed by all 5 authors. And so if you make a donation of $5,000 or more, you can have one. Now, you're going to have to move quickly here because I only have 10. So what you're going to have to do is email me at vincent@microbe.tv and say, I want to do this. And then I'll tell you how to send the check in. And then once you send your check in, that's one copy that's less available for others because I don't want to have people sending me checks and have no books for them. So 10 copies
Vincent,
only.
Print more. At that level, I would print more.
So I have a question. If somebody only wanted to contribute $4,000, would you rip out one of the autographs and then send it to them?
No, actually, if you'd only want to contribute $1,000, I'll just sign it. Okay, like last time, which a lot of people bought last year. I think we gave away 50 copies of the textbook last year. I thought that was impressive.
Wow.
Because you can buy it for $189. Right? So I will sign it for $1,000. All 5 will sign it for $5,000. Of course, my signature is unlimited, so there's no problem there. But I given last year's, I think this will be popular. So Vincent at microbe.tv, I have to get moving there. We need to raise money for this beautiful incubator and all the things that we do. I just talked to an audio editor yesterday who's going to help me edit the podcast, which I could really use. And so he's going to edit next week's TWiV, and we'll see how he said, I can make it sound better. I said, good. Make it sound better. Good luck. All right. So, Michael, where are you from originally?
Originally, I'm from Houston, Texas.
Wow.
And then I moved to upstate New York for undergraduate, and then moved to Manhattan for graduate school at the Icahn School of Medicine at Mount Sinai, I guess is now the official name.
Where in upstate New York?
That was at Skidmore College in
my
Saratoga
gosh.
Springs. Yeah.
Yes.
Yeah.
I
Really
remember
pretty part
Skidmore.
of the state.
I was at Cornell. We used to know Skidmore.
Yeah.
Nice racetrack also.
So then Icahn, and who'd you work with there?
There I was in Viviana Simon's lab at the Department of Microbiology. And I did my PhD thesis on the host restriction factor ApoBac3G, which takes out the lentiviruses, particularly HIV. And my work was really studying how the viral protein VIF specifically binds to ApoBac3G and the interaction between those two, how it evolves. And so then I moved to... Sorry, do you want me to keep...
I just
going
wanted
through?
to, I do want you to keep
Yeah.
going. I wanted to just say that I got my PhD at Mount Sinai also with Peter Palese. I was just there last week. And Viviana, of course, has been on TWiV also.
Mm-hmm.
Yep.
Yeah, I think actually all my mentors have been on TWiV, because then I went to Vincent Munster's group at Rocky Mountain Labs in Montana, and there I did my postdoc, really kind of building the research program that I'll be talking about a little bit today. And then from there moved on just kind of right across the mountains to Washington. So
How long ago did you move to Washington State?
I started in 2020, just in the summer of 2020, right as the pandemic was hitting. It was a fun time to try to start the lab.
I was just going to say, how was starting a lab during the beginning of a pandemic?
It was interesting. You really learn. I mean, and at the time I didn't have a technician, so it was really just learning supply chains and how to navigate just a very rapidly devolving situation. I remember like trying to get gloves. That was the first thing that I bought in the lab. And it was, I don't think I'd even started yet. Somebody at the school said the gloves are all running out, but you'll need gloves for your stuff to start. So let's just go ahead and buy gloves now. They might be there when you show up in July. So
it's challenging.
Been interesting. Yeah. Well, we got everything up and running in January of 2021 and really I think that's when the plate reader arrived, and then we could really hit the ground with our experiments. So it's been busy ever since.
So what got you interested in science to begin with? Do you remember?
Yeah, so when I was an undergraduate, I actually I went to undergraduate schools thinking I was going to be in theater. So I was looking, Skidmore had a pretty good theater program, and that's where I thought my interests were aligned. But I was taking some of the early science credits, and one of them was a neuroscience course. And a teacher emailed me. We were going back and forth over some quiz questions, and she said, "Oh, I can't. I'll have to get back to your question later. I have to go take care of the fish." And I said, "What do you mean by fish?" And she was running a zebrafish facility, so. At the time, she was also a new PI and looking for extra pair of hands. So, there was this kid emailing her about the fish, and kind of that was where I started, was in a neuroscience development lab looking at axon guidance in zebrafish. And that was where I really kind of learned, okay, this is probably more in line with my interests. And then I was also getting rejected from a lot of the theater programs I was applying for at the school.
But Michael, you've got the hair for theater still.
Yeah. Well, part of it's pandemic hair. It's just...
I like it.
You should have seen me when I started in December or so, 2020. It was just, I hadn't gotten a haircut in almost a year. It was touching my shoulders. And I remember the deans one day said, "Oh, I like talking with you. Your hair looks like it's exploding with ideas." And it's like, I haven't gotten a haircut in 8 months.
Is there any residue of the theater interest?
Yeah, and presentations. I think I do enjoy giving presentations a lot. I don't usually get too nervous making a fool of myself in front of an audience. So, presentations are right in my alley.
Yeah, that makes sense.
Michael, did you go to Rocky Mountain for Vincent's lab or for the mountains, or both?
No, A little bit of both. I do enjoy skiing. And there wasn't a whole lot of skiing in Manhattan, especially as a grad student. So that was very attractive. There was skiing about a half hour away from the labs, which was really nice. But it was really for Vincent's group. He, the intramural research program at the NIH, I'll just give a plug for that. It's quite nice because as a postdoc, I was given quite a lot of freedom to just go after the types of questions that I thought were interesting. And there are ample resources in the intramural program, which give the PIs a little more flexibility to kind of encourage exploratory research and then also encourage you to take it. So that was one thing Vincent and I talked about in the very beginning. That was a huge sell. He said, you can take what you work on here, what you make. So that was what really hooked me. And I think knowing that from the beginning, it really encouraged me to kind of develop my own kind of line of research and questions.
But you have to admit that Manhattan to Hamilton is pretty drastic, right?
Oh, yeah. Extreme culture shock. I mean, going from Manhattan to like a town of 5,000 people, all the restaurant, all the 1-2 restaurants that are in town and it close at 7. And so you really learn how to cook all the things that you need.
Houston to Skidmore is no cakewalk.
Yeah, right?
No.
But you can always go to Missoula for a good meal.
Yeah, it's true. And we did quite a bit.
What I wonder about is the switch from the neuroscience and the fish to the viruses.
Yeah, so in my senior year, I was taking there was a molecular biologist in the department, Pat Hillerin, that taught a virology course, an introductory virology course. And it was a night course. So we would roll in at around 7:00 in the evenings and, talk viruses for an hour and read papers. And it was a pretty informal course, but it was such a great introduction to virology. And it was, at the time I was also trying to figure out, I got into a summer undergraduate program at Mount Sinai, which is really quite a program. You can pick across a wide range of disciplines, and I was trying to decide which department to join, and all of these things kind of coalesced at the right time. So I went into microbiology. That was like, oh, okay, viruses are really cool, and I think I'm ready to try something a little different.
So I think early in 2020, I had known Vincent for a while. He sent me your preprint. This which subsequently was published in Nature Micro's Functional Assessment of Cell Entry and Receptor Usage for SARS-CoV-2 and Other Beta Coronaviruses. So it seemed like you were working on this, and then SARS-CoV-2 comes along, right?
Yeah. Actually, we were just ready to submit it, and then when the genome was released and we saw it was another Sarbecovirus, it was, okay, let's take another couple of weeks to finish this up. And it took us about 12 days, but we ran the full experiment and were able to show the receptor. So it was really just a drop into, I think, figure 3. But it was, it turned out to be quite an important little demonstration of kind of how quickly we could move in this assay.
Is it worth us going over this methodology? Because does this appear in the next paper as well?
Yeah, we do go over it a little bit. We do the same thing in this paper.
So tell us how you identify the cell receptor for SARS-CoV-2 in that first Nature Microbiology paper. How did that work?
Yeah, so really the question comes down to gene synthesis. It's very expensive to synthesize a whole spike sequence. If you want to test a coronavirus and you don't have the virus with you, you can synthesize the spike sequence, but that's over $1,000 and it takes a while. So one way we thought about doing it was to try to just do the receptor binding domain. But that on its own doesn't have a whole lot of function. So then you're limited to biochemical studies. Or if you can put it in the context of a different spike, something that you already have, then you can maybe test it functionally. So that's the approach we took. And we took the SARS-CoV-1 spike. We had it synthesized and made some silent mutations in it that allowed us to, at the DNA level, remove the region that encodes for the receptor binding domain. And then we could have a novel receptor binding domain sequence, which is only a few hundred amino acids, have that synthesized as DNA, and then we could clone it into the spike. And that whole process is relatively cheap. It's like $130, and it takes about a week or so to have everything synthesized. So it's kind of, it really changes the pace that we can work with these viruses. And importantly, what we generate out of this is a chimeric spike protein. So it has all the domains of a SARS-1 spike except for the receptor binding domain, which has now been replaced with whatever we put there. So we can do the SARS-CoV-2 spike and recover viral pseudotypes with this chimeric spike protein on the outside.
Yeah, so that was one of the things I wanted to ask. Do you put this spike on other viruses? So is it used for pseudotyping whatever is, you're not infecting with coronavirus, you're infecting with something else.
Exactly, yeah. Yeah, so importantly, this is all a biosafety level 2 kind of experiment. Mm-hmm. So it's all handled with a non-replicating recombinant vesicular stomatitis virus. So this VSV has been modified where they took out its own glycoprotein and replaced it with some fluorescent reporters. So it produces both GFP and luciferase, which is really kind of nice. We can easily see how well the experiment worked just visually with a microscope. And then if we choose to quantify it, we can go that route and add luciferase reagents.
So that's where the plate reader comes in that you were talking about. The plate reader arrived at your lab and you could do these experiments, right?
Exactly. Yeah, exactly. So as soon as we got the plate reader, then we could just start cranking through the assays.
And the challenge with making the full spike, as you mentioned at the very beginning, is just the size, right? Is that correct?
Yeah. And it's not necessarily a challenge. It's just more costly to have it all synthesized. Yeah.
Okay. So I ran into you and Brienne as well this summer at the BAT meeting in Fort Collins.
Right.
And you talked about work that's now been published in PLOS Pathogens, ACE2-dependent Sarbecovirus in Russian bats resistant to SARS-CoV-2 vaccine. So let's talk a little bit about that. And maybe you could give us your definition of Sarbecovirus to start off.
Yeah, so I guess we'll start big and then kind of work our way down. So there's coronaviruses. And then in the coronaviruses, there's different genera. So there's alpha, beta, gamma, and delta coronaviruses. The alphas and betas are probably the ones we're most familiar with. Those include viruses that infect humans and a number of other animals.
This is not to be confused with alpha, beta, delta, Omicron variants of SARS-CoV-2, right? My God.
Exactly.
Okay. This is
we need different
level.
Letters, I think. We need different Greek letters.
Yeah, I always tell my students that the problem is really just that we're not that creative. We go with sort of the alphabet that we use in English or the Greek alphabet, and that's all we know.
Sorry to interrupt. Sorry to interrupt, but I sensed a potential confusion there. So high level.
No, that's fair. That's good to point out. Okay, so then within the coronavirus genera, we have alpha, beta, gamma, delta. The beta coronaviruses are the ones that include the I guess fairly severe known human pathogens, SARS-1. SARS-2 and MERS. Mm-hmm. So the beta coronaviruses are kind of the human pathogenic, highly zoonotic coronaviruses. Within the beta coronaviruses, there are a number of lineages, or now they have different names, but you have they basically refer to groups of related coronaviruses, genetically related. So the Mirbecovirus is kind of prototyped by MERS coronavirus. And that includes a number, several hundred related coronaviruses found widely across the globe, actually. There's not really a solid one place. But then you also have the Sarbecoviruses. That includes SARS and SARS-2 and about 200 or 300 animal coronaviruses found in bats, largely in Southeast Asia. But now, as we've seen in the paper we'll talk about today, we also find them in Russia and Europe. And then there's several other groups of beta coronaviruses that are found predominantly just in wildlife, and they have different names, Nobecoviruses, and et cetera.
So Sarbeco is a mix of SARS beta coronavirus, right, basically?
Yeah. They're beta coronaviruses that are related to SARS.
SARS, okay. Got it. Now, furthermore, within this Sarbeco viruses, there are clades based on the receptor binding domain, right? So can you explain that?
Yeah, and that's something that we really kind of established in our 2020 paper, building off of some of the phylogenetic lineages that had come out. So Like I said, there are several hundred beta corona or Sarbecoviruses found in bats. And if you look at just the receptor binding domain sequence from all of these hundreds of viruses, there's about 30 different permutations of the sequence. This number goes up a little bit as we find more variants, but there's not hundreds of variants. There's tens of variants of the receptor binding domain. And you can think of them, they kind of fall into 3 groups or 4 groups, depending on how you want to think of them. But we call them based off of their sequence similarity to each other. So the clade 1 viruses include SARS and SARS-2 and a number of the bat-related viruses to those two. Those viruses have the ability to use ACE2, either from their own species or from humans. The clade 2 viruses have 2 giant deletions in the receptor binding domain relative to SARS-1. And those 2 deletions ablate much of the interface that we know with ACE2. So those viruses do not use ACE2 under any conditions that we've been able to test. And then the clade 3 viruses are kind of, and I talk about it a little bit later, but they're kind of potentially in between. They have 1 deletion, and they seem to be, what's now been shown is that they're fairly species specific for their own host ACE2. So they can use ACE2, but by and large, most of these clade 3 viruses are specific to their own, and they don't really cross, they're not really compatible with other ACE2s.
Is there a similar kind of pattern of evolution between them? Does it seem like one of those things arose first and then became the others, or does it seem to be independent evolutions?
Yeah, I think it's still up in the air. So there's, if you look at some of the work that comes out of Jesse Bloom's group and Kenneth Matriak's group, they've looked at a little bit more of these evolutionary histories for these viruses and how those traits might arise. And I think it's still, was it that it was gained over time, or was it a feature that was lost over time? And I think it's still a little unclear.
So
I'll just leave it at that.
The viruses the Sarbecoviruses that don't use ACE2, you don't know what the receptor is. Is that correct?
That is correct. And that is an active area of research in my lab right now. So what we showed in the 2020 paper was that there are a number of these clade 2 viruses that under the certain conditions do seem to be able to infect human cells. And so we're now trying to tease apart really that mechanism. And we have a paper coming out at the end of the year that it goes into more detail about what trypsin is doing and how that mediates entry and everything.
All right, so the next part of this paper are a couple of viruses from Russia. Tell us about those.
Yeah. So we did the first screening study in 2020 and have since then been using the tools to test new viruses as they come up. And so we tested some viruses from Uganda and Rwanda in an earlier study. And
these are
since I started my lab,
Sarbecos, right?
Yes.
Okay.
Yes. Yeah, these were Sarbecoviruses that were found in bats in Africa. So pretty widely out of where we expect them to see Sarbecoviruses. So as part of this effort, when I started my lab at WSU, we just hit the databases every week and look for new viruses that are discovered. It seems like, especially in the SARS-2 era, there's a new virus found every other day. So there's plenty to work with. And sometime in the summer last year, there were these 2 curious viruses that came up that were found in Russia called COSTA-1 and 2. And they're found in the horseshoe bats, predominantly on the western part of the country near Sochi National Park. So
is that where the Olympics were, in Sochi?
Yeah. Not too far off from where those were.
Okay.
But yeah, so as soon as we saw that those were Sarbecoviruses, it's a great fit for the system. And importantly, those were clade 3 viruses. So in our first paper, there was only maybe 1 or 2 clade 3 virus sequences in the database. So we couldn't really make a whole lot of strong conclusions there. And then the viruses we tested from Africa were also clade 3, and they also didn't tell us a whole lot. So that brings us to a total of like maybe 4 viruses that had been studied in this group. So any of the new ones that show up there are immediately of interest because they're just not tested very much.
So, excuse me, remind me again. The clades are defined phylogenetically by sequence. Is that correct?
Yes, but also by function, as we showed in our papers. So,
all right,
we showed that yeah.
And is the sequence phylogeny focused on the receptor binding domain, or are we talking about the whole spike?
No, that's another the way we do the phylogenetic analysis is just on the receptor binding domain and only at the amino acid level. So it's just giving us kind of an overall, more like sequence similarity and relationships rather than true evolutionary history that we can extrapolate from.
Okay.
Yeah.
Vincent, did we do this plus pathogens paper before?
Yes.
We did it as a preprint, no?
Yeah, because it was really familiar.
Yeah, we did, and then I know Vincent and I heard about some of this at the BAP meeting as well.
So you don't want to stop then, right, Rich?
No, because I got a gazillion questions. I'm holding myself
and also
back here.
we should call figure 2 the Rich's figure because it's only in here because of a comment you made on the episode.
Really?
So we have
right?
to get to that one.
Oh,
it's true. Yeah,
isn't that interesting? Because figure 2 part D today, I spent about a half an hour staring at that thing. Okay, because it's fascinating. I also want. We'll wait till we get to it. I got some questions.
All right.
Sounds good.
All right. So where were we? We're talking about...
the Costa viruses. Yeah.
Yep. Costa.
Yeah. So we saw that they were, as soon as the sequences were put online, we put them into our system. We had them synthesized. And at the time, I was working on a number of other projects in the lab. So I made pseudotypes, and I said, at the end of the summer, this is a good project for a student that's coming into my lab. So I'll just put these in the freezer. And then we didn't touch them for another several months. And then I think in November is when we did the first experiment with them and saw, that the clade 3 viruses one of them could actually use human ACE2. And that was really quite an exciting moment because it was really not anything that we were expecting. We were thinking, these are clade 3 viruses. Like, they've never done anything for us.
It's incredible how projects can mature with incubation in the freezer for several months.
Yeah,
it's really great.
It's true. It's true. So sorry.
I was just going to say, so Costa 1 and 2 of the isolates from Sochi, right?
Mm-hmm. Yep.
What are the clades again? Remind me.
They're both in clade 3. So
clade
these
3.
guys have one deletion and they're missing a lot of the contact, or generally the clade 3 viruses are missing contact points we don't see with ACE2. Generally. That's not what we saw here, but
I had a question about that. They're missing the amino acids, but does that mean that there's no contact between, the protein? Do you get what I'm asking? They have to contact something. They're not just out there in the air, right?
Right. So, that the amino acids that we find in the viruses that use or combine human ACE2 are not conserved in a lot of the clade 3 viruses. So, those amino acids have different identities, and as it turns out, those are probably also involved in interacting with ACE2, but just not from humans, rather with ACE2 from the bat species.
Okay.
So because the interface is just different enough from the bat side, the virus doesn't have mutations that can compatible.
These were found in horseshoe bats, did you say?
These were found in horseshoe bats.
All right, so you drop these Costa RBDs into your system, right? It's a it's a SARS-1 based spike.
Yeah, so we recover a SARS-1 spike with the Costa 1 and 2 RBD.
And then you do your plate reader, and what'd you find with Costa 1 and 2?
Yeah,
did we already define what's on the plates? Do we need to review that for the listeners?
Yeah, I was going to go through it.
Good.
Yeah, so we made our chimeric spikes, and then we generate these viral pseudotyped particles that fluoresce on entry, and then we plate out cells. In this case, we start with HUH7s, which are a human liver cell line. And we plate those out on the cell, on the plastic, and infect with our virus. And we do two conditions. So we include the virus with or without an exogenous protease called trypsin. And what we saw, I'll focus just on the kind of the without trypsin stuff. I think it's easier to kind of focus on that part of the story first. But what we saw is that under a regular, non-protease conditions, we could see that the COSTA-2 spike, or the RBD from it, could infect cells, could infect the HUH7s, and the COSTA-1 spike could not. So then we found that when we added protease, we could increase the entry of all of these virus pseudotypes in our assay. And the protease allowed for the COSTA-1 spike to enter, and the COSTA-2 kind of, a little bit less. So the point of the protease is to basically bypass another entry barrier. So spike is kind of this multi-step machinery. It can bind to the cell, it interacts with the receptor, and then it is processed by a host protease in the cell, and that allows for the virus to fuse. Unfortunately, a lot of cell lines are missing some of those proteases. So even though you can get a receptor binding domain engagement and you get the virus to attach to cells and et cetera, if the protease isn't there, then the spike doesn't infect. And one way we can get around that is if you include trypsin, then if there is still an interaction at the receptor binding domain level, then the protease can process the spike and the virus can infect. So this is one way that we kind of broadly enhance viral entry, and it allows us to detect kind of low levels of entry. So even if there's a slight interaction with that receptor binding domain and the receptor, the protease allows us to kind of get rid of whatever would quell that signal and let us actually kind of detect it.
So there's the requirement for trypsin, does that mean, do these cells have temperus too? These HUH7, do they have temperus?
I'd have to remember again if they. So the temperus, yeah, they do actually.
Okay.
They do have it, and then it's just low levels of the receptor in general. So if you look in, I think figure 1D, if you look in the untreated condition, the virus doesn't seem to get in very well. It's pretty low level across all of, it's like under 10-fold.
Yeah.
And then when we add the protease, the receptor is present, but the proteases are there in low levels, and that allows us to pull up the viral entry.
I mean, it's even the case with SARS-CoV-2 that if you add protease, you get a lot better entry of the pseudotype, right? So these cells are just not really good at...
No, they're not. They're like a first kind of try for us, because the HUH7s support a number of coronaviruses under different conditions. So we always test them first, or 293s or KCO2s, some of the very standard virology cell lines. And then because the result was interesting, we saw one of the viruses could go in and maybe one of them couldn't. Then we go after the more specific questions. Okay, which receptors are actually involved? So we change cell systems when we're looking at specific receptors. I don't know if you want me to just go into the next one, or
sure, go ahead.
Okay.
Yeah.
So this is a BHK cell line. Baby hamster kidney cells are pretty useful in our hands because they don't have receptors that are compatible with the human coronaviruses. So they might express low levels of their own receptors, but those are very generally incompatible with SARS-1 and MERS and SARS-2, et cetera. So BHK cells, luckily, though, are very transfectable, so we can give them a receptor in trans. We can transfect them with a plasmid encoding for ACE2 or DPP4. And so that's what we did. We repeat the same experiment with our pseudotypes, except now we're using cells that we've given different coronavirus receptors. And we choose 3 common receptors used for human coronaviruses. So we choose ACE2 because that's for the Sarbecoviruses. We choose APN, which is another receptor used by the by common cold viruses. Like we use a virus called 229E, which gives us the common cold. And then DPP4 is what the Merbecoviruses use, including MERS. So when we do this experiment, we saw just the COSTA-2 virus infecting. So the COSTA-2 virus infects, and the other ACE2 using viruses can infect with ACE2, but the other receptors don't work for the COSTA viruses. So it tells us that ACE2 is very much the receptor for these.
Do we have any idea about Costa 1?
Right. So yeah, Costa 1 is kind of like the running, kind of mystery in the background of this whole paper, because what we see is that Costa 1 really easily gets into the HUH7s with protease, but it doesn't get into the cells expressing ACE2. And we even try an additional cell line in figure 1G, where these are a human cell line, 293Ts, that are now stably overexpressing DPP4. So there's more ubiquitous protein expression. We're thinking we're giving the cells more ACE2 than a BHK transfected with ACE2. So you can try to give more ACE2 into the system this way, and we're still not seeing this Costa 1 virus going in. So it's whatever the entry that we're seeing with protease and the HUH7s is, and it's something we talk about later, and it is a segue into other work that we're working on, but this trypsin dependent ACE2 independent entry is a very interesting phenotype we're after.
So, I mean, the obvious explanation is that Costa 1 is binding to a receptor on the HUH cells that's not present on the others. Am I thinking straight?
No, that's exactly where we're thinking, but there's always arguments to be made either way. So.
Sure. Right, but it's not ACE2 anyway, right? It's
not
clearly
ACE2.
not ACE2. Right. Okay.
Right. Yeah. Right. And we even did an experiment where we tried to add protease, because I said that would increase entry. So we tried protease on the BHK cells with the ACE2, and we don't see the Costa 1 getting in. So it's just more evidence against Costa 1, at least in this set of experiments using the human ACE2.
Mm-hmm. Okay.
But Costa 2 definitely does. And I think that gets us into Rich's figure 2. Because when you guys first presented the preprint, I think Rich was the one who asked. He said, it would be really interesting to see what the structure would look like here, or what these contact points are. So I sat down, I think, right after the episode, finished playing on my computer, and had the structures, started downloading and modeling the structures. So in figure 2, we actually show the contact points that have been found in the crystal structure for SARS-1 and 2 with ACE2. And then we used Swiss Model, which is a web server that you can put in any protein sequence, and it will find crystal structures for the closest matching sequence to your input, and then it will generate and fit the sequence data from your sequence into the structural data. So it basically generates a crude model of the receptor binding domain for Costa based off of SARS-1 and 2 data. And that's what we have in panel C. And just showing the contact points that we find in SARS-1 and 2, those residues that are identical in SARS-1 and 2 are shown in figure 2C. Most of these contact points are conserved between SARS-1, 2, and Costa 2 with ACE2. So it's not too surprising why Costa 2 so easily uses ACE2. It's kind of the take-home point from this figure.
I was wondering about if you've used AlphaFold for any of this, and whether that's useful.
No, I haven't, but I do really want to get into the AlphaFold stuff. I'm very interested in applying that, I think, in the next set of papers.
I keep hearing that that's quite powerful.
Yeah, it does seem to be changing a lot of this structural biology game.
Okay.
All right, so then you say, next you ask, what about the full spike, right? Right,
and this is really to kind of still, I think, with this chimeric spike approach, one thing we're always trying to do is prove that it's a useful thing in the first place. So one way we can do that is to show that the chimeric spike does mimic what a full-length spike would do. It wouldn't be a very useful platform if the full-length spike was telling us something that the chimeric spike was telling us something the full-length didn't. So we spend the extra money and time, and we had the full-length spikes synthesized for Costa 1 and 2, and then we compared them in pseudotype assays just side by side with the chimeric spike. What we see, I think, is pretty consistent with what we've seen before. The chimeric spikes and the full-length spikes generally show the same phenotype. If the virus uses ACE2, in the chimeric spike, the full-length also uses ACE2. There is a difference. The chimeric spikes are a little bit more efficient. We see a little bit better entry with a full-length spike than the chimeric, and I think one explanation I have for that is that the SARS-1 spike is a pretty optimized human-compatible spike. So those other steps I was mentioning with protease processing and potential attachment and other factors involved in entry might be more efficient in that background when we consider just replacing the receptor binding domain. So generally, the SARS-1 chimeric spikes kind of give us a best-case scenario of the entry that we'll get with the receptor binding domain. The chimeric spike shows the same thing, but just a little bit less.
All right, and the last part of this paper. This is probably the part that scared people, right?
Well, it wasn't really supposed to be. I mean, it's still always within the context of pseudotype.
Sure.
So that's always important to remember.
So tell us
but
about
really,
that.
Really, the concern is that we're seeing SARS-2 spill back into wildlife. So it's been found in the deer population on the East Coast. It's found in minks, in pets, cats. It's kind of a concern that we see this virus so widely distributed, people now able to get back into the animal population. So what we know about the animals are that several of them can carry their own Sarbecoviruses, or at least their own coronaviruses. The Costa-2 Sarbecovirus is kind of a worst-case scenario, but what would happen if SARS were to get into a host population that has some kind of Sarbecovirus like the Costas? So to mimic this kind of scenario, we took the same approach of making a chimeric spike, but we changed the background. So now it's a SARS-CoV-2 spike, and we're replacing its receptor binding domain with Costa. Ultimately, what we'll test with it later, and the last figure of the paper, is how well this chimeric spike is resistant to vaccines and therapeutics. But what we show first off is that the chimeric spike is able to infect cells expressing ACE2, so the SARS-1 and SARS-2 backbone don't really change the story there. And then to get into testing, we tried incubating these pseudotype particles with bamlanivimab, which is one of the first monoclonals that was released under emergency use for SARS-2. Importantly, it's a monoclonal directed towards the original isolate of SARS-2, the RBD from that virus. So this is not too surprising when we make pseudotypes that have that exact RBD. That antibody can bind to the spike and basically prevent those particles from getting into the cells. So that's what we show in 1C. The chimeric spike with the Costa-2 RBD is not very well bound by this bamlanivimab, and it isn't reduced. So we don't see very much reduction in entry when we do those things. So to then test for vaccine efficacy, we collaborated with Bronwyn Gunn, professor right down the hall from me here at WSU, who has a collaboration with Tulane University, basically for patient samples for COVID screening. And we were able to get serum from individuals that had received 2 doses of either the Moderna or Pfizer vaccine. So we combined those sera with our pseudotypes and tried to infect our ACE2 expressing cells. And what we saw was that the SARS-2 full-length spike was nicely inhibited by serum from people who had received the vaccines. But again, the Costa-2 chimeric spike was notably reduced in its ability to be inhibited by the virus. So it seemed to be somewhat resistant to at least that kind of first 2 doses of the vaccine in this assay. So when that was originally where the paper ended, and we submitted it in April. And over the peer review process, that was just enough time for Omicron to emerge. So one of the reviewers then came back to us with, well, how does this resistance compare to Omicron? So we made another chimeric spike with an Omicron RBD and repeated the experiment. This time we also got a hold of some serum from individuals who had received both vaccine doses and then had acquired Omicron through a breakthrough infection and had recovered. So we had the serum from those individuals. And so we could see again that the just standard 2-dose vaccines don't inhibit even the Omicron RBD. Only the serum from the patients that had the breakthrough infection could reduce the Omicron RBD from infecting cells. But the COSTA-2, again, it was reduced more this time, thankfully, by the Omicron infection. But it still wasn't as striking as what we saw with the actual SARS-2 Omicron spike. So COSTA-2 is still more resistant, I guess, than the Omicron variant, which is not too surprising because, I mean, as we've seen, even across the evolution of SARS-2, alpha, beta, and the delta and Omicron variants, just slight amino acids in the receptor binding domain give a huge result in the vaccine efficacy. So when you really have what is essentially a very different spike here from COSTA-2, it's not surprising that the vaccine doesn't work very well. But I think it's more the kind of the overall message of, well, you know, what are the viruses then that we're including in the universal vaccine design? And are we including some of the clade 3s? And I think, thankfully now, it seems to be that several groups are now including some of the clade 3 viruses, or really just trying to broaden their design to include more coronaviruses.
You know, this mosaic nanoparticle strategy of Pamela Bjorkman, are they including clade 3s in there? Do you know?
Yeah, I think actually I was talking to Vincent about that because he's on that paper, and it does seem like that is at least a direction that they're going in.
Okay, good.
So do we know anything about the COSTA viruses themselves not using pseudotypes? I guess I'm thinking about my teaching, and you show that they're susceptible, but do you know that they're permissive or if human cells?
Right. No, very little. Although I will say that in general, with these coronaviruses and beta coronaviruses, kind of as it seems, the spike is the predominant barrier for these. So even some of the viruses that don't infect human cells very well or at all, if you replace the spike or even the receptor binding domain from SARS-1, for example, the virus can infect cells. So it argues that much of the machinery in the rest of the coronaviruses is compatible across a wide range of species barriers. You can get a bat coronavirus. If you change the spike to something, you can get it to replicate in a BHK cell, a hamster cell, basically. You can get it to replicate in a human cell or in a primate cell. So I'm not sure how big the barriers are past entry, but we have not tested anything full virus. And actually, as far as we know about the distribution of this virus, it's pretty limited. So, how often are people going to come into contact with the COSTA virus is also unknown at this time.
So the sequence identity, I don't know if it's identity or similarity between COSTA-2 and SARS-2 in the RBD is about 60%. Is that right?
Yeah.
So they're pretty dissimilar. And yet, looking at the structural modeling, the critical residues for maintaining ACE2 binding, they may not even be in exactly the same place in the sequence, but in the structure, they come out so that you can maintain the ACE2 binding. So it's as if you can change a whole bunch of stuff around that to the point where it's no longer recognizable by plasma, but still does the same job. Is that right?
Right. And I think that's what we've seen. SARS-2 is a good example of that. SARS-2 has quite a lot of variation along those contact points that other viruses would make with ACE2. But those contact points themselves are conserved. And I think, as you point out, it's the variation outside of that really drives the resistance to, drives resistance to cross-reactivity between the viruses.
Interesting. Wow. Okay.
I think one of the concerns you bring up is that these COSTAs or similar viruses could we combine with other coronas, right? And then there you have the ability to infect cells, and the antibodies aren't doing anything, right?
Right. Yeah, and this is a kind of the long-running problem with coronaviruses is that there is so much of this kind of crosstalk and recombination in the natural host reservoirs, especially for the Sarbecoviruses. There's a wonderful study from Zhang Lixi's group in 2017 showing an entire cave where every animal that they sampled has a slightly different Sarbecoviruses, and you can imagine just the enormous amount of diversity and recombination that occurs there.
So I have a question, because it seemed to me, correct me if I'm wrong, I think in the 2020 paper there was one clade 3 virus, and now 2 years later, in this new paper, it looks like there's one that you're calling clade 4. We haven't even mentioned clade 4, this RATG15. So will there be 5 clade 4s in 2 years? Or
who knows? Yeah, the clade 4 is kind of it's a newer virus that was described last year by the Wuhan Institute of Virology, and it's just dissimilar enough where the phylogenetic trees are placing it out of it doesn't cluster as closely with the rest of the ACE2 using clade 1 viruses. But it also doesn't really cluster with SARS. It really does form its own type of separate branch. So I think right now, even though it uses ACE2 and it behaves very similar to some of the other clade 1 viruses, we're not entirely sure where to put it yet. And I think when we get more sequences, then it'll help us better readjust our trees. I'm working on one now, for another phylogenetic tree for one of the next papers that's coming out. And it seems like every time I make one of these trees, the topology changes just a little bit because we're stacking in more sequences that kind of change a little bit of our understanding.
So there's oodles of coronaviruses out there, but we actually know the receptor specificity for only a handful of these. Is that correct?
Yeah.
And so we have a tendency to focus on ACE2 or other known receptors and say, oh my God, there's an ACE2 binding virus out there. This is a threat for a spillover. But hey, there could be any number of receptor specificities out there that could spill over that could use a human receptor that we just don't know about. Is that right?
Mm-hmm. I think my favorite example of that is this virus called HKU4. It's found in 2006 in a big screening study, and it was just put in a database and nobody cared about it. Then MERS coronavirus spilled out in 2012, and groups started looking through the databases retroactively, and they found this virus HKU4. And then when they started making pseudotypes, HKU4 can also use human DPP4 and the receptor that we now know for. So, 6 years before the human virus emerged, we already had a virus in the database that had the compatibility with the human receptor. We just didn't. But because there was no MERS virus that was on anybody's radar, nobody would look at those viruses and say, oh, this is something we need to consider.
But your system provides the potential to screen RBDs out there quickly to see what other viruses might be out there that could recognize even an unknown receptor on a human cell, right?
Yeah, that's the advantage to doing the functional kind of the chimeric spike approach. As I was saying, if you only have the RBD sequence, you can go after the question biochemically, but that requires you to know the receptor or the binding partner to do your test to see if there's compatibility. So by doing a more with like kind of more black box, you can actually get more at the biological question of does the sequence have a zoonotic risk without having to know from the host end.
Right. So if you want to ask that question of all these sequences out there, which of these might, based on the capability of binding a human receptor, pose a zoonotic risk? What do you use for cells?
Yeah, so I think that's a huge part of these platforms, the type of cell choice. So we're working on the Merbico type platform now to go after the same question, but for the MERS viruses. And a huge amount of the effort has actually turned into what cells to use. I spent years working on various prototypes for this assay, only to find out that nothing was working because I was using the wrong cell line.
Right.
So I think there's probably not one single right answer for the cells, and it is kind of a battery of different cell types to use.
I wonder if you could even
which is what we do.
I wonder if you could use a mixture somehow for a quick screen.
Yeah, that's we actually have some triple receptor cells in the lab that express APN, DPP4, and ACE2, so we can kind of overexpress everything and see if there's just basal compatibility, and then from there weed out just the top hits and see which ones are specific for which receptors.
So I finally have a question here. Are there any bat cell lines that you can develop to make sure that you're looking at the right host?
Absolutely. So, yeah, that's actually an effort in my lab. So we do develop bat cells, and we have about a half dozen brand new ones that we've been kind of characterizing for the past year, doing just that, checking for kind of compatibility, not just with the Sarbecoviruses or even the coronaviruses, but we screen a number of other viruses in the lab for compatibility, and really just trying to build new tools, build new bat lines that allow us to test some of these types of phenotypes more, specifically. Are there bat cells right away? There are some. We did test a few in the 2020 paper. They're not of much utility. Most of the time, the bat cells, for some reason, lose receptor expression in culture, so they usually need extra coercing to be susceptible. But it is definitely something that we are actively working on.
Are these mostly lung cells, or are they from other organs as well?
No, a wide variety. So we have brain, kidney, lung, liver, and spleen.
Wow.
From some species that we're working on.
Okay. Thank you.
So I wonder if you're willing to mention the IMPRESS study that you did with the Wuhan Institute of Virology. I think that's really quite interesting that you could even do anything with them, right?
Yeah. It's been interesting. Definitely, it's a collaboration that I've always wanted to do because they are responsible for all of the Sarbecovirus knowledge that we have in the field. Predominantly, all the bat coronaviruses have been discovered by TWiV, so it's just an incredible, talented team of bat virus researchers. But yeah, it's so that work is actually looking at this ACE2 independent, Trypsin dependent entry that we talk about. So everything that we've done up till now has been with pseudotyped particles and single cycle entry. So can the Trypsin conditions that we show in some of these papers, and we show it with Costa 1 here in this study, but we showed it with a number of the clade 2 viruses in our first study from 2020. Is that actually a real phenotype? Can that support multiple cycles of replication, or are we just kind of forcing viral particles into a cell with too much trypsin? And so I collaborated with TWiV over the past year now to test recombinant coronaviruses. And what we found actually is that the conditions that we outlined in the first paper and we go through in here and the Plus Pathogens paper actually do support some of these clade 2 Sarbecoviruses that have double deletions. So really showing the conditions needed for this group of viruses to replicate. So that's kind of that paper in a nutshell, really looking at what is it that a Sarbecovirus needs for our viral culture assays.
So before we let you go, Michael, maybe you could just tell our listeners what should they take away from this? What are some of the main takeaways that the person on the street should know from this, say?
Well, I think that coronaviruses are widely dispersed. So for a while, I think, at least, especially in the field, before we really started to go out and find these viruses elsewhere, it was a Southeast Asia issue, or maybe just, China at first. And then as groups started to look in their own backyard a little bit more, they start to see, okay, we also have Sarbecoviruses. Stuff that is still, it's not SARS, but it's still 70% similar. So the fact that we do have these viruses more widely dispersed is one point, and that their phenotype, the ability to infect human cells, that is, it's not just that they're emerging, that they could just emerge from China. It's not just that they could come to us from Southeast Asia even. We find Sarbecoviruses, now the clade 3 viruses have been found in the UK, Poland, Russia, across Africa. So I think it's, not to say that these are going to emerge necessarily, but just when we're trying to figure out where these viruses come from and we talk about origins, I think that we just haven't really learned enough about nature and what's circulating in our own backyard to really call a lot of this yet.
Are there Sarbecoviruses in the Americas?
Not that we've found yet. I don't think that there's a whole lot of surveillance there, but not yet. Unless you want to include SARS-2 that's spilling back into the deer.
No, I don't want to do that. Well, is it a matter of not having the right bat species, perhaps?
Perhaps there is a really big study about a year and a half ago looking at ACE2 compatibility across 40 different bat species, and there is some compatibility there. So,
okay.
It's lower though. It's lower.
The other thing I got from the TWiV collaboration, you make a statement that viruses in most field samples can't be propagated. What we have are genome sequences. We can't grow them up, right? That's an impediment.
Yeah, it's a huge, and I think that's there are a number of coronaviruses that even infect people that haven't been able to grow. So HKU1, it was notoriously considered an unculturable coronavirus, and it wasn't until groups figured out you needed primary human airway epithelial cells to get them to grow. So it's not that the virus doesn't work. It's just we don't know what it takes to grow. And lots of people have tried to grow the coronaviruses that they sequence out of samples, and most of them don't grow. So it seems that, and that some groups have even tried cells from the species that the viruses were found in. Especially TWiV has tried for years to rescue some of these viruses in Rhinoceros cell lines that they have, primary or immortalized, and it just doesn't matter. Just the cells aren't there, and I don't think our knowledge is there yet. So,
yeah. In fact, go ahead, Kathy. Go ahead.
No, go ahead.
The famous RATG13. No one's been able to grow that virus, right?
To isolate it. I mean, we can get the spike to work in cell culture,
right. but not the entire virus. Yeah.
Right.
So, Michael, you just answered my question because David Quammen a couple of weeks ago told us how, as an autodidact, one problem he has is that oftentimes he sees words before he hears them. And when I saw the bat genus that you just mentioned, I pronounced it in my head the same way you did because I thought it should be kind of like rhinoceros. It should be Rhinolophus, right? Yeah, but then I heard lots of people saying Rhinolophus. So now, I'm forever trying to figure out which way to say it. But but you said it Rhinolophus. So
I said it right. I can't say that I'm the expert on how to pronounce the word, though.
Yeah, but it is pronounced both ways, and I did hear it a couple weeks ago somewhere, either someone reading David Quammen's book or someone on TWiV said it Rhinolophus, and then I thought, okay, so the way I saw it first is possible. So just curious.
Rhinolophus or Rhinolophus, huh? Interesting.
Rhinolophus. So with all the interest in bat research surging, I would say, and everybody's now wanting to get on the bandwagon, are there colonies of bats that have been established at laboratories, at universities? And then if there are, and if you're dealing with an exotic bat, is there not a chance for an escapee or two? And that's what are the biosafety hazards about just keeping bats?
Right. So when I was at RML, I was involved in a bat study at BSL-4, where we were infecting them with Nipah virus, and that was, I mean, it's BSL-4, which is its own extra level of biosecurity, but bats are generally not the easiest to handle.
Exactly.
So they're either yeah, exactly, and there are only so many people at RML that were small enough while still in the inflated bubble suit to be able to climb into a large bat cage and grab the bats. So that was my job there.
Do the bats? How much room do they have to fly around? Are they kind of doing the metabolism and flight that they would do in nature?
It depends on so in a BSL-4, it's very restricted, and it's not the most ideal situation for that. But in larger, it just depends on the bat itself. Some of the smaller bats you can get away with a little bit less space, but yeah, generally they're not getting the most for some of these types of studies, not getting the most natural environment. It's a pretty sterile, weird place to bring a bat.
When I was
lab.
when I was at the bat meeting, after the meeting, Tony Shouts took me to see. They have a colony of Egyptian fruit bats, I believe.
Oh, my.
And they're not very big, maybe 6 inches, and they're being bred in a room which I would say is 10 by 10 feet. And there's 200 or 300 bats in it, so they're flying around. And she said, "Be careful when you first go in; they may pee on you." But and then if they want to infect them, they take them and put them in smaller cages, as you said, because you can't be flying around and being infected, right? So,
Wow.
but we heard
there are colonies, that get to.
Okay.
Yeah, there are colonies of bats that are available for some research.
I mean, one of my favorite exhibits ever is at the Bronx Zoo. It's called The World of Darkness. And if you go inside that exhibit, you can even see fish-eating bats, because they actually have a couple in there. It's an amazing display, but I often wonder, what do you do with the bats now that you don't know that when they get old and they have to go to bat heaven, you don't want them to die on the premises? So do you release them back where you caught them, or what happens?
I can actually I can tell you what happens. They go to me, and then they become cell lines. So that's. So some of the cell lines that we are generating in my lab are actually from WSU's bat colony at their Vancouver campus in Washington.
So they donate their bodies to science.
They do.
They can.
And these are what, little brown bats mostly, or some common bat that's found?
Yeah, that's a Carolia perspiculata. That's just a kind of a mid-sized bat, but it's predominantly, I think the colonies are predominantly driven by what animals will breed in captivity and are easiest to handle. Rosettas are some of the more standard, I guess laboratory bats. And then beyond that, it's pretty diverse depending on who's building the colony and what their needs are.
Right.
Yeah, we heard some good stories at the bat meeting, if you remember, of people capturing wild bats and bringing them back to Berlin, right? Remember that one?
Yeah.
Wow.
And the airlines said, no more. We're not doing this. We're not doing this for you anymore. But I don't even remember what kind of bats those were. I forgot. Anyway, Michael Letko, Washington State University, thanks so much for coming and joining us today. It was a lot of fun.
Yeah, thanks for having me.
This is great. Thanks, Michael.
Thanks. Yeah.
Thanks. It's really cool.
Okay, so I want to go to Pullman, Washington. I've been sitting here. I've been multitasking here, looking at Google Maps. It's just 15 minutes away from Moscow, Idaho.
It's gorgeous.
It's about an hour and 20 minutes south of Spokane. I have opportunities to do this because I go through that country occasionally when we drive either to Seattle or Oregon. It'd be worth a detour. The other thing I need to do is to go to the Bronx Zoo. I've never been there.
Oh, no kidding.
And it's right next to the Bronx Botanical Garden, too. Right?
It is, which is lovely.
And I've never been there either, I go to New York. It's what a heck I am. I go to New York and say, what am I going to do, right?
Oh, come on.
But I need to go to the zoo and the Botanical Garden. What a great idea. All right.
Yeah, the zoo's good. Dixon and I and Daniel went to the zoo. Remember, Dixon?
I do. Looking at penguin malaria.
That was a lot of fun. They have a great resource, too.
The
laboratories are amazing.
Zoological Society is there, I think, also. And when I was at Rockefeller as a postdoc, bat echolocation was a big topic at Rockefeller. And there was this guy, his last name was Griffin. I think it's Donald Griffin, I'm not sure, but and he gave talks on this that were absolutely fascinating. You just didn't know that the extent to which wildlife can behave, and that was one of his points. I think that Michael had a good point. We have no idea about the behavior of these animals, and whether they do at night, they're not telling us, and we're sound asleep. So, apart from the vampire bats that we know about from the movies, that's about it. I mean, the secret life of bats is really largely unexplored. largely unexploded. Hard to do also.
All right, let's do a couple of emails here. Rich, can you take that first one?
Sure. Abigail writes, good afternoon. First, thank you for all the information you so freely share for those of us who are information junkies, but are desperate to know where to find accurate evidence-based information. Your podcast, along with the Unbiased Science Podcast and your local epidemiologist's blog, have been my go-to resources during the pandemic. I'm grateful for the time you put into sharing science and making it accessible. I want to start by saying that our family has been very COVID conscious. My children, ages 7 and 11, wore masks at school all day, every day, from fall 2000 through spring of 2022. Until recently, my husband and I masked at work and indoors in crowded spaces. We were vaccinated as soon as we were eligible in early 2021, and our children received their vaccines in November and December 2021. We have all also been boosted, again, as soon as we were eligible. My husband is in active military duty and has a vaccination record that is a mile long, and that's not great hyperbole. Only my husband has ever tested positive for COVID, but that was in Iraq, and he had no symptoms, 2020. But I admit that I have had a lot of pause about getting the new bivalent booster. I have received 3 Moderna shots, and the second and third made me horribly ill. I did not have the typical flu-like symptoms. Instead, I had an excruciating headache accompanied with vomiting for about 48 hours each time. I was completely unable to take care of myself, much less for my children and my home, and subsequently, I felt off for several weeks. I'm loath to attempt that again, especially with voices like Dr. Offit's positing that benefits for me would be short-lived. I also admit that my anxiety is fueled a bit by reading this study, and she provides a link, and the accompanying anti-vaccine, pro-ivermectin, anti-establishment comments made by doctors on Sensible Medicine. This is Science Direct. Okay. If adverse effects are occurring with such frequency, and at greater frequency than some other vaccines whose usage was discontinued, this is referring to this article, the risk does not seem worth the benefit. What I would love from your hive mind is an assessment of the data. I do not believe in some great conspiracy, but I do wonder why we do not hear more about the risks of these new vaccines if the incidence of adverse effects is so great. Thank you, Abby. Okay, so I had a quick look at this paper, which is in Vaccine, dated September 2022, titled Serious Adverse Effects of Special Interest Following mRNA COVID-19 Vaccination in Randomized Trials in Adults. And what this does is it's essentially a review that it's more than a review, because it goes back and it sort of reanalyzes a lot of the available data on adverse effects from the vaccine trials for both the Pfizer and Moderna vaccines, and has sort of a closer look in particular at what they call adverse effects of special interest. I have to say, I did not look at it in sufficient detail to be able to offer my own independent assessment of the data. Superficially, it looks to me like the increase in adverse effects in some of these instances that they're talking about are pretty small. And they also have a huge number of qualifiers at the end of the paper that, to me, the bottom line is they're saying this might be worth looking at in some more detail. So I don't see anything in this paper that's going to tell me that, these vaccines are dangerous in a fashion that has previously been unrecognized or anything like that. I would want to look at it in more detail and maybe have another real expert opinion on that. But I didn't see anything there that immediately caused me great concern.
Yeah.
I have two other quick comments. One, this is anecdotal, but a person known well to me had the same sort of problem with, I believe it was the first boost, yes, or the second immunization, the second shot of Moderna. Not exactly the same symptoms you're talking about, but really totally wiped out with what appeared to be effects that lasted for weeks, and struggled with what to do about the first boost, and ultimately did the Pfizer vaccine because it's a lower dose and maybe has a slightly different adverse effect profile. And in that particular case, once again, this is anecdotal, had no problem at all. Okay, so it could be that choice of vaccine makes a difference. That having been said, yeah, we have a lot of experts who are saying, if there's no crying need for the boost, you might be okay without it.
Yeah, I think the two things I notice is that she's talking about getting a fourth shot here, which there's definitely less data on than a third. And I guess that's all I can say about that. And when I look at the paper that she has linked, my read on it is it is talking about, exactly as Rich said, looking in detail at the serious adverse effects that happened in some of these trials and sort of trying to quantify and talk through exactly what there is. But I don't see data here really making any comparison with other vaccines. So the idea that this is worse than other vaccines that you mention here is not supported in this paper, or is not presented in this paper. And I don't see data on that. And if I was going to try to really think about that and understand it, I would be concerned about making sure I had studies in similar populations. And so if they're looking at historical studies, or if someone else is making that comparison, I would ask a lot of questions about the populations in whom those adverse events were studied. This paper does not have those data talking about comparisons with other vaccines. And they certainly haven't done a head-to-head in similar populations and similar numbers of patients and things like that, which is what I would really want to see if we were going to make an argument about if it is actually more than in other vaccines.
One thing that's at the end of the introduction, which you can think of sometimes as an expanded abstract, is that they say, our analysis is restricted to the randomized trial data and does not consider data on post-authorization vaccination program impact. So they're working from a smaller data set, and kind of what Brianne was just alluding to, that might be a particular population type that, and so their next sentence is, it does, however, show the need for public release of participant-level trial data sets. So that would give the data, I think, that Brianne's asking about.
I also noticed that they conclude in the Moderna trial, the excess risks of serious events, 15 per 10,000, was higher than the risk reduction for COVID-19 hospitalization relative to the placebo group, which was 6 per 10,000. And it seems to me that that's not a fair comparison, because you have all these serious adverse events, which, some are less serious than others. And it doesn't seem to me that comparing them to hospitalization is the right way to do it. But I'm going to run this by Paul Offit, see what he thinks.
Good idea. The paper.
Yeah. I want to add my 2 cents here, Rich. You got an N of 2 now instead of 1. I had the Pfizer vaccine for the new booster, and a person very close to me had the Moderna. And this person, who is still very close to me, was sick for 3 days, had a big red wheal, which didn't go away for a week and a half. And the same exact thing happened down the hall to another couple. So I have a feeling that the Moderna vaccine, although it is very effective, has a bigger kick to it and has more side effects.
In some people, let's qualify it that way.
Of course, not everybody. Yeah. Not everybody. That's a fact. Yeah. And that was even true for the initial vaccination schedules. That is.
Well, among other things, it's a higher dose. Plus the composition, the lipid composition and stuff is slightly different. So.
Yep.
Yeah.
Kathy, can you take the next one?
I think so. This is Visto.
Yeah.
Yes. Okay. Sorry, I was looking at the paper. Visto writes TWiV 944 with Catherine. That's not spelled right. Hanley. Was very interesting. So eloquent, adventurous, and knowledgeable she is. Borneo fieldwork does not sound like the cushy academic work environment. She mentioned that VSV, dengue, and other mosquito-borne viruses overwinter somewhere unknown to reemerge the next summer. Could it be that we could eradicate some vectored viruses by removing the vector? I'm thinking of gene drive technology that can eliminate a species of mosquito from the environment. Because I am not Dr. Evil stroking a white pussycat, I propose that a virus-free population of the temporarily extincted mosquito species be kept in the lab for later release. Would breaking the transmission cycle for a year or two have a chance of succeeding in eliminating some of these vectored viruses? And Visto gives a reference to a Nature paper, which I haven't looked at yet.
Basically describes the technology about behind the gene drive and propagating mutations in a mosquito population.
Right.
If we could eliminate mosquitoes, we would. That's been a big difficulty, and I don't give any hope at all for completely eliminating any vector species. Not at all.
So Dixon, what sort of ecological consequences would there be to eliminating mosquitoes?
Well, if you want to do that experiment.
A lot of what? He's
frozen up.
Oh.
Too bad. I was interested to hear what he was going to say.
Vincent, Dixon, you back? No, he's still frozen up. All right.
I guess there'd be consequences, right?
There would definitely be consequences, but the idea of at least saving some virus-free population for possible later release seems like you might help to mitigate that. Not a bad idea.
Okay. Brienne, can you take the next one?
Sure. Neva writes, Hi, Twivissimos. The confusion about the two Sean Carrolls was understandable. Since I am familiar with both, let me tout the physicist Sean M. Carroll. There is plenty of online bio you can check out. Having followed him on the webs for years, I especially look forward to his Mindscape podcasts as he interviews a wide variety of folk from different fields. Recently, he moved from Caltech physics to be Homewood Professor of Natural Philosophy at Johns Hopkins University in Baltimore. His interview with biologist Sean B. Carroll is in Mindscape number 117 from October 2020. Look at the list of other interviewees, and I'll bet you'll find some that interest you. He, like you all, is wonderful at and devoted to explaining science clearly and with good humor. Many aspects of physics, especially particle physics, just dizzy my brain, but much of it is fascinating. His Mindscape website is https://www.preposterousuniverse.com. Yes, the universe is preposterous indeed. As Vincent said about viruses, you just can't make this stuff up. His latest book, The Biggest Ideas in the Universe, can be a TWiV pick if you like, for the curious who are okay with a bit of a challenge. I am enjoying it very much. TWiV has been hitting it out of the park even more lately. Thank you, Neva from Buddha. And she notes, P.S. time for another lunch on the patio, Rich and Ibby.
That lunch has already happened. That's a dated email.
Oh, okay.
Did you go to Buda or?
Yeah, we went to Buda. Went to Garcia's restaurant in Buda. You know, What a history. What an amazing history. Neva dates back to well pre-Austin for me. And, you know, those early conversations on, is it pronounced Buda or Buddha? And all of this kind of stuff. Great stuff.
All right, I'll take the last one here by Michael. Hello, I'm an avid TWiV listener and a historian of modern and contemporary art. Given how often you discuss vaccination, particularly in the clinical updates with Dr. Griffin, it occurred to me you might be interested in this piece I wrote on what is arguably the most important and widely recognized modern image of vaccination, a panel by Diego Rivera in his series of murals in Detroit. Provides a link to the painting Vaccination, 1932. Wow.
Mm-hmm.
This is really, I've run across these murals before. I've never seen them in the flesh, but a really quite famous assortment of murals.
Mm-hmm.
Vaccination is one in a cycle of frescoes that Rivera created for the Detroit Institute of Arts, adorning the walls of the museum's garden court. Have you seen those, Kathy?
Yes.
Cool.
The name of the mural is Detroit Industry, and
Nice.
it's huge. It's a highlight of the DIA. Yes.
The unnerving resonance of Diego Rivera's Vaccination, Art Forum International. And Dixon put a little note, outstanding. Over the course of the past couple of years, I've kept coming back to the image of Diego Rivera's Vaccination. I can't get it out of my head. The primary reason should be fairly obvious. This is arguably the most iconic and widely recognized artistic treatment of a subject that many of us have been reading, talking, and thinking about incessantly during the pandemic. But there's more to it than that. If conventional accounts would lead us to expect mural art to be direct, didactic, and declarative, Rivera's image is anything but. Its effect is subtly disquieting. It gets under your skin, measured. Gosh, she's definitely an art writer, huh? I noticed the pictures by Keith Haring behind Amy during the most recent live stream, so she might be interested in this art history angle as well. Michael is a professor of art history at Hunter College here in New York City. Hello, Michael. Nice to hear from you, neighbor.
Yeah.
Yeah, I got to say this picture is subtly creepy.
Mm-hmm.
But I'd have to see it in the context of the rest of the mural because it, the style generally is, well, I don't know if it's creepy, but it's certainly unique.
Yeah.
And this article this article actually continues on with several other illustrations of vaccination far out. This is a pic, dude. I need to read this. Holy cow. This is cool.
Brianne, what do you have for us?
So I have an article in New York Times Science that someone sent me, and I really was amused by it.
That's hilarious.
And so I had to pick it. The title is "Sometimes This Octopus is So Mad It Just Wants to Throw Something." And it is actually talking about a study that was published on the gloomy octopus and some behavioral studies that show that it apparently can sometimes live too close to other octopi in an area of Jersey Bay, Australia, that is known as Octopolis. And the octopi actually have been shown to have a behavior where they throw silt at each other when they are exasperated at one another. And apparently this is something that is not common in the animal kingdom. But at the top of the article, there is actually a video of one octopus throwing silt at another. While at the bottom of the article, you can see another video of an octopus discarding trash. So you can see the difference in the motion and the difference in the behavior of an octopus just throwing something because it doesn't want it versus this actual exasperated behavior. It was just really kind of fun to watch. Also, whoever wrote this put captions on some photos that were very funny captions. So I laughed harder than perhaps I should have when I was sent this article and thought that everyone else might want to see it. And then when I looked at the papers we were talking about today, I was reminded of an astronomy picture of the day from Halloween. That was one of my absolute favorites that I've ever seen, called the Cosmic Bat Nebula. And it is in fact a nebula that looks like a bat.
Okay,
and
cool.
I thought that was just the greatest ever. And it does tie in with today.
It's great.
Yeah. When I saw that one, it didn't make an impression on me because I saw it from too close. But someone was standing in the doorway of my office and they said, "Oh, wow, look at that. It's a bat." And then I saw it. But otherwise, it looked kind of like a lot of the other, pictures.
Yeah.
So, yeah.
Nice. Thank you. Kathy, what do you have for us?
Well, I picked a trifecta of astronomy pictures of the day. So the first one is Jupiter, and it's a YouTube video. And I have to say, I think Jupiter is about my favorite planet. So I really like this one. And it has a nice music background. Sometimes, videos will have an annoying music background, but this one I found kind of soothing. And then the next one was a picture that I have to admit, I don't quite understand the image itself. It's 3 pictures from the lunar eclipse that was this week. And I did get up, I woke up and I said, I could go see if I can see the eclipse. And I could see a big bite out of the moon. And but I didn't see anything like this. So this is pretty cool. And then the one that's for today is called Blood Moon Ice Giant. And with all this talk about the eclipse, I didn't realize that there was also this thing that where during totality it passed in front of, or occulted is the actual word, outer planet Uranus. And so there's a picture of that really brings it home. And then, of course, I had to look up the pronunciation of the name of that planet. And if you Google for pronunciation of Uranus, it talks about various things, but one of them is that astronomers tend to put the emphasis on the first syllable, Ur, and then say Unus. But then there's a thing that says it was changed to Uranus in 1986 or 85 when one of the space probes was preparing to do its flyby. Newscasters around the country realized that 3 weeks of Uranus would never work, especially when also tossing in the reference to a deep space probe.
That's great.
So anyway, if you believe the internet, astronomers prefer Uranus.
Uranus, that's just great. Deep space probe.
Yeah.
I love it. Rich, what do you have for us?
So I have a pic that was sent to me, an article that was sent to me by Justin, one of our correspondents, called Mirror Image T7 Transcription of Chirally Inverted Ribosomal and Functional RNAs, which is kind of a mouthful. And unfortunately, it's behind a paywall. So I'll just, it's the overarching concept that I want to communicate here, because this is downright weird. I've also linked to a wiki article called Mirror Life that overviews this whole thing. So as some of our listeners may not appreciate, the building blocks of both proteins, that is amino acids, and nucleic acids, that is nucleotides, their chemistry is asymmetric, so that in fact you can isolate mirror images of these, okay? Because there's at least one asymmetric atom in the middle of each of these. And interestingly, in nature, both proteins and nucleic acids are each built of just one of the possible mirror images of all the amino acids or nucleic acids. Those are L amino acids and D nucleic acids. And theoretically, you could do the whole thing with the mirror image amino acids and nucleic acids. Why is another question. This paper has, in the discussion, so basically the paper is about a step in creating essentially a mirror image central dogma of molecular biology, okay? Which would entail having LDNA that you can replicate and LRNA that you can transcribe from the DNA. And if you could make a mirror image ribosome that would have the mirror image amino acids and nucleotides to make up the ribosome, that thing could theoretically make D protein. All right. And interestingly, as you might have guessed, in order to replicate LDNA, mirror image DNA, you need a mirror image protein. And in order to transcribe mirror image RNA, you need a mirror image RNA polymerase, okay? And so in this paper, what they've done is to create a mirror image T7 RNA polymerase that they show can transcribe mirror image DNA into mirror image RNA. That's just insane. The real challenge, if anybody, I guess, they're pretty invested in this. My guess is they'll go for it eventually. I mean, creating a mirror image ribosome, what a task.
Wow.
Because you're talking about, 40 or 50 proteins, depending on whether you're eukaryotic or prokaryotic.
Yep.
They got the RNA part figured out here because they've figured out how to make the ribosomal RNA. But once you got the system established, it's interesting because it's sort of creating life from scratch, right? Because once you got the system established, then you're okay. You can do the whole thing. The big question for me is why?
Right.
What they say here in the first part of their discussion is the realization of mirror image T7 RNA transcription may enable a variety of practical applications of high-quality long LRNAs in diagnostics and therapeutics, and they give references, information storage, computation, imaging, and basic RNA research. Furthermore, the ensemble of efficient mirror image DNA polymerase, RNA polymerase, reverse transcriptase, and sequencing tools may lead to the realization of a mirror image selection scheme for the directed evolution and selection of LRNA aptamers, targeting biologically important molecules as potential clinical and research tools. I don't have the vision to really appreciate that. I'll trust them on this. But the technology and the concept I find at least amusing, if not fascinating.
Amazing. It's just amazing.
I've posted in here a meme that
I love it.
my
Really
students really like, and it helps them understand this very well, of a picture of Samuel L. Jackson and Samuel D. Jackson to help one remember and understand what the difference between L and D is.
I hope this goes chiral.
And that the meme says, I hope this goes chiral.
That's great. Yeah. I love it.
This leads me to expect that in the mirror image world, everything will be reproducible. Okay. Yes, this is part of one of the things they say is if you had, for example, I get the enantiomers mixed up. If you've got mirror image RNA, it might be more stable. Okay, for all the therapeutic values, therapeutic uses you would use RNA for. But I question whether it would also have the same reactivity. Okay, is it going to do the same thing? I don't know. They do talk about aptamers. Okay, which suggests that at least some of the binding properties of the mirror image nucleic acids might be similar, or I don't know. But at any rate, your question spawns that response. That's I would expect, yes, that the mirror image proteins and nucleic acids would be more stable because they wouldn't be degraded by the enzymes that would work on the natural product. Yeah. So, yeah, I suppose, actually, if this is interesting, if a mirror image protein does the same biochemical reaction, and you want to, if you can somehow get that made or introduce it into an organism, then rather than getting turned over, it might hang around for longer. Maybe, I don't know, and do the reaction you're interested in. I don't know. Check in 20 years from now. maybe.
This also comes up in a science fiction book that I've read.
Okay.
And there, it is very important that some food is from the D enantiomer, and so you cannot get calories from it, but you can just get the taste.
Oh,
Ah,
okay.
interesting.
I'm not convinced you'd get the taste. You need a mirror image tongue to do it.
Yeah,
Right, taste it.
I couldn't tell you the details, but that was part of the science fiction book.
That's cool. All right. My pick is an article in Science-Based Medicine by Clay Jones, who's a pediatrician. It's called The 2022-23 Respiratory Virus Season Hits Early and Hard, Part 1. It's about the high levels of respiratory virus infections this fall. But this one focuses on RSV. And we've heard about a lot of RSV and influenza and SARS-CoV-2. But this one I like because he explains the consequence. So he says, I've been a pediatrician since 2003. Never have I personally experienced a month like the one we were having. RSV had always kept us busy, most common reason for infants to be admitted to a hospital. But I have never seen admissions come in at this place. He works in Boston. He says, what happens if a child is in the ER, is in the emergency department, needs to be admitted to an inpatient unit, but there aren't any beds available? Well, they can be transported from the ED to a pediatric inpatient bed at a different facility. Easy, right? Not so fast. What if there aren't any pediatric inpatient beds available at any nearby facility? No big deal. You can just transport them to a facility that's not so nearby. And in some cases, these children are being moved hundreds of miles away from where they entered the system. And if there are no beds to be found, the child has to board in the ED, sometimes for days, waiting for a bed to open up. None of this is ideal. It increases expense and, more importantly, the chance that a mistake is going to be made. It gets worse. Not only are the numbers of children requiring admission historically high, these patients are trending sicker than usual. They don't just need supportive care for 2 to 3 days. Normally, they usually just need supportive care for 2 or 3 days with IV fluids, oxygen, perhaps a brief period of feeds through a nasogastric tube. And then he goes on to talk about these kids. But I thought this was good because, we talk about viruses and antivirals and vaccines. But he says, he gives you some details which are really quite interesting.
Wow.
Yeah.
He works in a decent-sized community hospital with a 12-bed pediatric unit. We've been at 100% capacity for weeks, managing 20-plus patients, some days 75 to 80% with RSV. Our ED is struggling to keep up. So this is very good to see what's going on.
Yeah, they announced on the radio this morning that the hospitals in Michigan are full. Our hospital is full.
Amazing.
Pediatrics. Yeah.
Wow. I'm waiting for the messenger RNA RSV vaccine. Okay, RSV's been a real problem. And it seems to me that the messenger RNA technology might be an approach to this. I'm just looking at the Moderna site. They have an RSV vaccine in like phase 1 or something like that. I'm surprised there isn't something more advanced.
The news reports, have mentioned at least 4 things. Go ahead, Brianne.
Yeah, I was going to say, I knew I had seen it, so I just Googled it. And there are 2 RSV vaccines that are in phase 3 right now.
Are they mRNA vaccines or other than that?
It does not look like it.
Okay.
No, it looks like they're recombinant protein.
Okay. Unfortunately, we have that vaccine debacle.
Yep.
Okay.
Mm-hmm.
Yep.
With enhanced disease that really, we'll see how it works out.
This does say that Moderna has a phase 3 mRNA.
Yeah, that's what I thought. It doesn't show it on their pipeline thing, but good. All right, we'll see.
We have a listener pick from Debbie. Is a fan of TWiV for nearly 3 years. I'm sending you a link to my favorite science show on Canada's CBC radio network. Bob McDonald has hosted Quirks and Quarks, For 30 of its 40 or so years on the air, this celebratory episode gives a lovely overview of who he interviewed, the subjects he has covered, and how this show has inspired people. Bob will continue as a science broadcaster. It's not a retirement show, and I recommend it to anyone who is curious about science.
Cool.
With much appreciation for all you do to educate us about the science of very little things called viruses. It's a good way to put it. Very little things called viruses. I like it. It's TWiV 954. Show notes, microbe.tv/TWiV. You can send your questions, your comments, your picks, your praise, your criticism, whatever you'd like. TWiV at microbe.tv. And if you enjoy our work, or at least appreciate it in some way, please consider supporting us. Microbe.tv/contribute. Dixon Despommiers at Racaniello.org, thelivingriver.org. Thank you, Invisible Dixon. Brianne Barker's at Drew University. I don't know, you still on Twitter, Brianne?
I am still on Twitter right now. Yeah.
All right, BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here.
Kathy Spindler's at the University of Michigan in Ann Arbor. Thanks, Kathy.
Thanks. This is a lot of fun.
Rich Condit is an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough, always a good time.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. By the way, Jolene has now got her own lab at Texas A&M. She says it's just getting going and really exciting. So good luck. Good luck there, Jolene. What did I say? I said I was Vincent Racaniello, right? You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.